AIDS stands for acquired immunodeficiency syndrome, and the main causal organism that causes AIDS is human immunodeficiency virus or HIV virus. So let's just look at what is AIDS. So we know that our body is protected by our immune system. So the immune system is our defense system of a country. What AIDS simply do is weaken our immune system. And once this defense system of a country is weakened, it is susceptible to terror attacks. Similarly, our immune system, when it is weak in case of HIV infection, it is susceptible to infection by various other pathogens. Now, the main cause of AIDS or the main cause of transmission of AIDS is sex without a condom or sex with multiple partners. So it is transferred by bodily fluids like semen, etc. And also it could be passed from mother to its baby. Sharing injecting equipments, needles, etc. can also cause AIDS. But what is the misconception about transmission of the disease is it can be transmitted by insect bites, toilet seats. No, it can't be. Because insect bites, toilet seats, kissing, etc., is not causal of transmission of AIDS. And in a separate video, I'll be discussing all these topics in details. But let's just look at the rates at which AIDS is growing day by day. At least several countries all across the world is heavily affected by AIDS, and one of the topmost of the in the list is Africa. And also India is a high risk zone for AIDS propagation. Now the main causal of AIDS is the HIV virus, or more popularly known as HIV-1 virus, because there are two type of virus, HIV-1 and HIV-2. Now HIV-2 is kind of restricted to specific West African population, whereas HIV-1 virus is one of the most common viruses that is detected from AIDS patient sample. Now HIV virus, let's just get a description of HIV virus. HIV virus has its GP120 surface glycoprotein, which is important for the viral entry into the host, and we would look into the mechanism in details. It has its matrix proteins, it has viral genome, which is basically an RNA, and it has all the viral enzymes, which comprises of integrase, reverse transcriptase, and protease, which has defined function in terms of viral propagation and generation of new viruses, which we would be discussing shortly. Now, the first step of viral entry is binding of the viral surface glycoprotein GP120 to the CD4 of the T cell. So the T helper cells are the primary target of HIV viruses. Imagine the HIV virus is just like a terrorist. It is attacking our body and making the soldiers as a hostage, because CD4 T cells are the soldiers of our body. Now they get inside the body of these soldiers, or otherwise the CD8 positive T cells, and grow inside them, and in a process when they buds off, they can even destroy the CD8 positive T cells, and as a result they cause severe harm to the immune system, because the number of CD4 positive T cells is reduced drastically in AIDS. So the first step is membrane fusion. So frankly speaking, the viral GP120 exists as a trimer, and apart from GP120 there is another thing. The stock is made up of GP41. This whole surface glycoprotein binds to the CD4 co-receptor on the T cell. Now, before interaction with that CD4, the interaction domain for the CCR5 co-receptor on the T cell surface is hidden inside a pocket, which is revealed outside when it is interacting with the CD4. Now, the interacting domains, which is going to interact with the co-receptor, is revealed, and now it can interact with the CXCR4 or CCR5 co-receptor, and as a result there is a fusion of the virus with the cell, with the CD4 positive T cell. This would allow entry of the viral genetic material inside the T helper cell. And along with the viral genetic material, integrase, protease, and reverse transcriptase also get inside. First, the reverse transcriptase would reverse transcribe the viral RNA to a DNA, and this process is known as reverse transcription. And after the reverse transcription, the DNA would be getting inside or integrated into the host genome by the enzyme integrase, just like the name suggests. So after the HIV genome is integrated the host genome, it would be transcribed just using the host transcription machinery. And these HIV genome contains several gene segments. Important genes comprises Pol, GAG, and envelope. The Pol gene encodes for these three important enzymes, that is reverse transcriptase, integrase, and protease. We already discussed about the function of reverse transcriptase and integrase. Let's just talk about protease, because protease is also very important for viral life cycle. Protease cleaves several polyproteins, which is important for viral matrix or the viral glycoprotein or the capsid assembly. So we take an example of GAG gene. The GAG gene, which is also known as group specific antigen, codes for the precursor GAG polyprotein. Now the GAG polyprotein is a huge polypeptide, which is in turn cleaved by protease, and that gives rise to several capsid and matrix protein, which would constitute the viral matrix and help to build up the viral particle. Other than GAG, envelope is coding for GP160. GP160 is a precursor for GP120, which is further cleaved by protease to give rise to GP120, which is the main surface glycoprotein important for HIV viral entry into the host cell. So that is how we understand the key function of the genomic segments of the HIV viruses, the GAG, Pol, and envelope. Other than these important genes, they also have VPR, NEF, VIF, PPU, etc., which all kind of help the virus to disable the immune system or the detection of the virus by the immune system. So they have varied function. And after all these things, all this processing is happening, the viral particle is getting assembled onto the membrane. The newly synthesized genetic material is getting incorporated, and a new virus is ready to bud off from the surface of the CD4 positive T cell. And in the process of budding off, it generally don't harm the T cell, but sometimes when the viral load is increased, too much budding off from the CD4 positive T cell also kill the CD4 positive T cells and hampers its normal functionality. So in that respect, it can kill the CD4 positive T cells, and there is a drastic reduction in the CD4 positive helper T cell population in HIV/AIDS patients. Now, here is an overall summary of the process we have discussed till now. But what is the treatment of the AIDS? What is the diagnosis of the AIDS? Now, about diagnosis and treatment, I have separate videos. But how can one stay healthy when it has AIDS? Now, there is a social enigma that when you have AIDS, you are kind of isolated and something like that. That gives a mental trauma. So AIDS is normal. It's like it's a disease just like other viral diseases. So there is nothing to worry about, and you can also make your friend or the person who is affected making more friendly by interacting with them, and also taking the medicines, which are a combinatorial therapy that I would be talking in another video, can help to keep the increase the lifespan in case of AIDS. And otherwise, taking balanced diet, proper exercise, and mental support can help the patients to live longer when they are affected by AIDS. So if you like this video, give it a quick thumbs up, and don't forget to like, share, and subscribe. And also comment on the video to tell me that how do you like my video and what improvement you want in these videos. Thank you.